Good evening, students. Myself, myself, Dr. Vinod Kumar. I did my PhD from National Institute in Biotechnology. So today I am going to teach you about Hybridoma technology. Hybridoma technology. It's a topic of biotechnology. So it's the main use of this is to generate monoclonal antibody. Generate a monoclonal antibody. So what is a polyclonal antibody and monoclonal antibody? When antibodies, so before going to differentiate between monoclonal and polyclonal antibodies, we need to know about antibody and antigen binding sites, the interacting site. So the place where the antigen bind to the antibody is what the places are called as, or the sites are called as. There is one terminology called TOPE. So epitope and paratope. There is a terminology called TOPE. It is of two types: epitope. The other one is paratope. So epitope present on the antigen and paratope present on the antibody. So if we generate a antibody which is more specific to a single epitope, then it is called monoclonal Antibody. Then it's called monoclonal antibody. So if we generate, topic is Hybridoma Technology. Need to generate monoclonal antibodies. Monoclonal antibodies. Those are more specific, more effective. More effective. So why did they turned it as hybrid? Because it's a use of two cell lines. So one cell line is B cells, which helps in producing antibody, and the other cell line is myeloma cell, which are cancerous cells, immortal cells, and continuous growth. So by using this two characteristics, that is one with continuous growth and the other with antibody production, they make a mAbs. So they injected mice with to isolate B cells. They inject mice with antigen and collect the B cells that produce antibodies. And at the same time, they collect the cancer cells and modify them to collect the cancer cells and modify to produce modify to make it lack of HGPAT engine. HGPRT engine. It's most important. So in this, they fuse two cells. B cells and myeloma cells with the help of PEG, that is called polyethylene glycol. So with the help of PEG, they fuse both the cells. So once they are fused, they should grow in a separate media, that is called as AD media, hypoxanthine aminopterin thymidine. So this media components have a specific use, that aminopterin helps in the blockage of the enzyme that helps in DNA synthesis, nucleotide synthesis. Aminopterin blocks the enzyme called dihydrofolate reductase in the nucleotide synthesis. Dihydrofolate reductase. Reductase enzyme is blocked by the aminopterin. Basically, nucleotide synthesis happens in two pathways. One is salvage pathway and the other one is de novo pathway. So this aminopterin blocks the de novo pathway. De novo pathway is blocked by aminopterin. So this hybridoma technology uses fuse of two cells, B cells and myeloma cells, in the presence of PEG, polyethylene glycol, and they are grown in the hat medium, which has a specific use. Hypoxanthine, aminopterin, and thymidine is the full form. Aminopterin blocks the dihydrofolate reductase of de novo pathway. So de novo pathway is blocked. The cell should undergo the salvage pathway. So in order to cells to undergo the salvage pathway, should need hypoxanthine pyrimidine. And one more important thing is HGPRT enzyme. This all leads to conversion of nucleotides for DNA synthesis. So once we grow the cells in hat media, there is a possibility of getting 5 types of cells. One is the myeloma plus myeloma, and next one is B cell plus B cell. The next one is unfused B cell, unfused myeloma cell, and the last one is hybrid cells. So usually the B cell's lifespan is very less. So that's the reason we are combining it with the myeloma cells. The B cell's lifespan is less. So they die fast. Myeloma cells, as they lack HGPRT, so they will die. And unfused B cells, less lifespan. And unfused myeloma cells, no HGPRT, so they will die. So only the fused cells will survive. Why they will survive? Because the fused cells have HGPRT from B cells plus they having the long lived characteristics from myelomas cells. So they are long lived and they can survive even in HGPRT enzymes by using this hypoxanthine and thymine and HGPRT enzyme. So they can synthesize only the hybrid cells can synthesize the DNA through salvage pathway. So only the fused cells survive. The help of hypoxanthine, thymidine, HGPAT. Continuous growth from myeloma cells. The next challenge is to find out which antibody is more specific towards the antigen in the binding efficiency. So next challenge is to find binding efficiency. Effectiveness. So for that, ELISA they will identify. So ELISA is nothing but enzyme link immunosorbent assay. Usually antigen is coated and ELISA is nothing but how strongly it is binding. We can identify protein-protein interactions or antibody-antigen interactions. We can identify based upon the more effectiveness and more binding efficiency. We can isolate that and we can use it for therapeutic purpose. Thank you for listening and watching. Please support, subscribe, like, and share. Thank you. Thank you very much. Please like, subscribe, and share.